Hello, everybody. I hope everyone is staying safe during these strange and peculiar times that we are living through. I'm glad that you were able to make time out of your day in order to watch my ISM presentation. Today in this presentation, I will be going over my experiences through ISM, including my first year in ISM, what I learned this year with my mentor, who I will get to in a second, and of course, I will be having you solve a murder. So let's get right into it. One of the first things my mentor ever told me, we sat down and he said, there's a very important quote that summarizes forensic biology in an amazing way. And the quote is, when two objects come into contact with one another, there's always an exchange of material. That's from Edmund Locard. And that is the principle of what I was studying in ISM this school year, was the transference of genetic material in the cases for criminal court and for other scenarios. Now, some of you are probably asking, what is ISM? ISM is an acronym, actually, and it stands for Independent Study Mentorship. Now, those are just three words. You don't know what those mean. Independent, meaning that the students are on their own. They have to schedule things. They have to make sure that everything is as it should be. They are not given, hey, you need to do this and this and this. You are given one paper a week, a grading cycle, saying when everything is due, and it is your duty to get that in on time. That's the independent portion. Now, study, it speaks for itself. You find an area of study in which you are highly interested in. For instance, it could be anything from magic to guitar playing to actual high-level jobs such as law, the sciences, oncology, a ton of varying fields. And you're probably asking, well, that sounds pretty cool. How do I do that? Well, the only thing you really need to do in order to do the study of your choosing is to find a mentor. And that's the last letter of the acronym for ISM, which is mentorship. We as students meet with our mentors once a week, sometimes more often, to discuss certain subjects, to talk about things we discussed the previous week, stuff like that. And it is their duty as a mentor to teach us about their job. For them, it's a job. For us, it's an area of study. So this year in ISM, I chose forensic biology. For those of you who may know, I did forensic psychology last year, which was a soft science, and this year's a hard science. The difference between those is a soft science is more theoretical, evaluative, and doesn't really rely on physical data. You evaluate somebody based on their mental capacity, and then you write a paper about that. Hard sciences is highly based off of evidence and physical data. So any science such as chemistry, physics, anything like that is considered hard science, and forensic biology most certainly falls into that. In ISM, we do these speeches, which you are currently watching. Usually they are in person, but for safety reasons, we are doing them online. One way that we get so great at speaking in public is through a process known as Toastmasters. Now, it is not what you're thinking. It's not about making a really good French toast or how to evenly toast a bagel. It's nothing like that. Toastmasters is a program in which they aid you to become a master of giving toasts. So if you've ever been at a wedding, you've seen the best man give a toast, or if you've been at any public event, there's usually someone speaking. And so Toastmasters allows us to go through certain, I guess the correct word would be practices? That's a very loose word. But we are given two things. One is prepared speeches, which are things like this, slideshows that we have had in advance and we have prepared and rehearsed and such. But the other portion, which most people find to be the most fun part of Toastmasters, is called table topics. The way table topics work is that there is a table topic master. The master chooses 3 people every session or week, or however your Toastmasters is set up. They choose 3 people and they give each one a separate prompt. And the prompt can be anything, absolutely anything. Please explain how the exact way the Berlin Wall fell, or tell me how gravity works in water differently, or please explain to me why Charles Dickens was not a great writer. It can be anything. And the point of this is that people who are called on by the table topics master, they are given this prompt and they have no clue what it is before they hear it. They are not told beforehand, hey, you're going to be talking about Charles Dickens or anything like that. Essentially, it's improv. And so it gets us comfortable with those areas in speeches where it's not scripted. Maybe the slides don't work, or you have to make something up. If someone asks a question and you're not 100% sure, it helps with that. As I mentioned before, I have a mentor. That's the third acronym in this, the third letter in the acronym in this class. My mentor is Mr. Garen Foster at the Bexar County Crime Laboratory. There's a little picture right there. I try to get more pictures of the laboratory, but as you can expect, it's a government facility with very confidential and sensitive information sprawled across it. So I don't want anyone's genetic profile to go on the internet by accident. So I was only able to take a photo from the outside. But Mr. Garen Foster has been a wonderful mentor. At the time of this recording, actually, I just got through with my final meeting with him, and I can honestly say he has taught me so much, and I am so absolutely grateful for having him as a mentor. And he's done the 3 main jobs perfectly. That's why I'm so happy that I had him. The first job, obviously, is mentor meetings. Those are essential for being a mentor. You have to meet with your mentee or your apprentice or whatever you want to call us once a week, if not more. And if you have to cancel a mentor meeting, then you have to work it out with your apprentice and make sure that you get maybe 2 meetings in next week, or balance it out in some way, shape, or form. The second one was hands-on training. And the picture that I just showed you is the front of the lab. I went inside an actual crime laboratory and did a multitude of experiments identifying blood, looking at sperm and egg cells, going into different rooms with very, very expensive and powerful UV lights that could only run for about 3 minutes before overheating. That's how powerful they were. And looking around for any stains or anything like that. And it really put things in perspective because I got to use so much equipment, so many chemicals, and it was amazing. It just put everything in perspective for me, and I'm so glad that I was able to do that. And thirdly, the third job of a mentor is providing research and direction. So sometimes I'll get an email from Mr. Garen Foster, and it will say, usually either a quote or a joke, just him being him, and then it will have some documents. So I am currently enrolled in online classes for crime scene analysis and bone collection and a bunch of stuff that when I was studying it in school, people would look at my laptop and see sketches of, grave sites and putting bones back together, and I had to explain what was going on. And he always, whenever we were going off track or he didn't feel like we were going at the right pace or something like that, he would always bring us back and say, hey, maybe we should focus less on this and more on this. And I'm so happy that he did that. I meet with my mentor roughly once a week. And I say roughly because there have been times where I've met with him twice a week. And there are times where I just see him because he is one of my best friend's fathers. That's how I got to know him. I met him as a freshman in high school, and I actually went on a trip with my best friend and Mr. Foster and Mrs. Foster. I went on a trip with them to East Texas, so I knew them extremely well. So when I called up and said, hey, I need a mentor, and you are in a field that I enjoy, would you mind taking an hour out of your week to talk to me? He said, of course. I also do lab visits. Lab visits are great. I go once every month or so, usually around once every 2 months. I've been to the lab twice, and due to COVID-19, I wasn't able to go anymore because they shut it down completely to the public. So we would go in, and it would be like a mentor meeting, but it would be hands-on stuff rather than talking about it. So then, rather than him sketching in a journal and trying to show blood cells and stuff like that, we would just pull up a bloody rag from the closet and then put it under a microscope and look at it. And that was amazing. Now, the title of this presentation probably hooked you guys in, and I told you I was going to help you solve a murder. And I'm going to do that by teaching you what I was taught over the year by Mr. Garen Foster. So how do you solve a murder? Right now, I probably would ask you guys, what type of things do you expect in crime scene solving and stuff like that? But I can't do that. So there are more than what is on screen. There are so many branches of forensic science, but these are the main ones that we are going to be talking about. There's blood samples. Blood samples are the bread and butter of forensic biologists. They are wonderful sources of genetic material, and they are usually very easy to spot. It's hard to miss glass that's broken and it has blood on it. It's very hard to miss. Genetic studies, that is looking at your genes, which we're going to be talking about in a second, mom and dad, and giving you chromosomes. That's the next portion, which is chromosome analysis. It's looking at your chromosomes and determining things about you through that. Again, we'll get to that in a second. Statistics. Whenever a forensic biologist or any forensic scientist goes into court, they usually have to have a strong case. And by a strong case, I mean they say there is a 1 in 14 billion chance that this person is not your criminal. And by doing that, it makes the case more solid. Again, we'll get to that in a second. And finally, criminology and victimology. Unfortunately, we will not be able to do this. This was a in-person thing, but we will still be able to look into the mind of the criminal and help understand the victim as well. So I said earlier, chromosomes, genes, and statistics. And you probably were like, oh, I remember that freshman year in high school. It was the beans and the monk, and he grew the beans, and they did different things. Well, the way it works in forensics is that we get sets of data, as you can see here, and by doing analysis on that data, we can move closer and closer and closer to the final data that would go to court. Usually that final data has very strong arguments and is scientifically proven and such. So, how do we look at blood, genes, chromosomes, and say, you robbed a bank? How do those two things correlate? And it's by STRs, another acronym, and it stands for short tandem repeats. What I mean by that is on screen you can see a double helix of DNA has been unraveled and split in half. And you probably remember back in high school or middle school, GTCA. Those are the 4 things that make up everything. Those are the 4 things in DNA. And so here we represent those by colors. And G goes to C, A goes to T, and they always match. What short tandem repeats are that is when these letters repeat significant amounts and are located in areas where not everyone has the exact same amount of repeats. So on screen there's GTAT, GTAT, GTAT 13 times. That would be a 13 STR. So that could mean maybe you have green eyes, or maybe you have double joints, or something like that. It's not things such as you have hair, or you have an eye. We look at areas that are very defining, because that allows us to narrow it down to that 1 in 14 billion that I was talking about earlier. And it shows up when you put the blood sample into the machine. It shows up like this as a graph. And this is very complicated to look at, but at the very top you can see D3S1358. And that basically is an address. That's saying it's DNA. Which chromosome is it located on, and which locus. So you can think of it as DNA is the country, which chromosome is the state, and which locus is the city. And that is a very simplistic version of what those numbers mean. And I said GTAT, GTAT, GTAT 13 times. Here we see 15, 16, 14, 16, 24, 26, and so on. Those are the numbers I'm talking about, and they're represented graphically here. So we can see in the DNA of the third chromosome, which is singular, at loci 1358, this person has a 1516. What that basically means is they got 15 or 16 from their mother and 15 or 16 from their dad. Genes and biology, which we all learned in middle school and high school, you get one from your mother and one from your dad, and that becomes your gene. So here they have a 15 and a 16. Well, that could mean something like they have blue eyes. And we've just by that fact alone, we have narrowed the possibilities down to a very small proportion. I believe 1/4 of the population has blue eyes. That means that we have excluded 75% of people on the planet, which is really, really good. So now we move on to 1416. Well, maybe that means they have, let's see, double joints, something like that. Well, 1 in every 100 people are double jointed. So if you take that 25% that we have earlier, then do 1/100th of that, you have an extremely small number of the population, and you keep going until you basically narrow it down to one person. And the easiest way to narrow down at least half of the population is by looking at the X, Y chromosomes. This tells you gender. So if for this instance, this says XY, which means this is a male. That means that 51% of the population has been excluded from being the donor of this genetic material. And again, you would go through all of this, input it into the system, and it would come up with a very, very small number that only one person on the planet could have been the donor of this blood or whatever genetic material it is. Here is the machine that does this process, as shown by Riley Foster. The blood samples go through the needle-like protrusions at the top, and then through 50,000 volts is pushed through the wires into a laser, which actually color codes the blood. As it's moving through, it's acting as though it's a filter. So small genes are moving through, and the bigger ones will come later. And that is inputted into the system, and it gives you the graph that we saw earlier. Now, if you would be so kind as to look into the description of this video and click the link that says Genetic Profile Maker. I'll give you a second to do that. All right, great. You have your genetic profile. You might have noticed the genetic profile is a card. It's not the graph that we saw earlier. And we're gonna solve a murder in very simplistic ways using that card. So, there has been a murder. We need a murder in order for this process to work. Who did it? Everyone is a suspect. All of you are suspects. So, we will be using, rather than CODIS, which is a system in which the genetic profiles input at the city, state, and national level, we will be using the computerized official frequency filing export engine, otherwise known as COFFEE. So this is the murder weapon. Very scary looking. But now we have put the blood sample that we found into COFFEE, and it gave us this, a very, very simplistic version of the graph that I showed you before. So I want you all to look at your card again, just remember what it was, because we're going to go through this. They, at this specific locus, it could be anywhere, they're a 4-7. Well, in real life, that could mean, again, blue eyes, double jointed, brown hair. It could mean a multitude of things, but in our scenario, it means their card is red. So that means, in a very gender-like way, like I spoke before, 51% being excluded by the donor being a male, we just excluded 50% of the deck, if you will, by stating that the donor of this blood sample has a red card. So if you have a black card, that means you didn't do it. If you have a red card, you are still a suspect. Now they have a 15-6, pardon me. That could, again, it could be you have a sixth toe or something strange. But in this scenario, it means that the card is a diamond. So first we said it was red, 50% gone. Now we're saying it's a diamond. So if your card is a heart, you were previously a suspect, now you're not. We have cut, in just 2 genes, we have cut the number of suspects down to just 25%. And when you're talking in populations where there are millions and millions of people, that means a lot. That means so much to cut down to 10%, 5%, even 20% is still a large cut. Moving on, they have an 8-10. In this scenario, it is not a face card. Aces, however, I am not considering faces. So if you still have an ace, you're still in the game. A 3-4, it's an odd number card. So if you are a 2, 4, 6, 8, 10 of diamonds, you have just been excluded. So now we are left with ace, 3, 5, 7, 9 of diamonds. That is 5 cards out of an entire deck. That is an extreme cut in the population. Now here it says 172. Now I told you before, these represent what you got from your mom and from your dad. And the third one does not mean that one of your parents was cheating. That's not what it means. It means that there was contamination. Maybe some dirt got in the way, or maybe the CSI was not extremely careful, and maybe their saliva got on the blood, or something happened where the genetic material for this specific gene was contaminated. So we are not able to use it. A 5-2, this means it's sharp. This is where we get specific. In gene testing, you can get extremely specific. It's how your body is made. And there are many details of which your body is made. So sharp meaning that it is Now an ace, a 5, a 7. So 9 and 3 were just excluded. So now we are left with 3 cards out of an entire deck. A 2-8, it means it has a letter. So let's review. It was red. It was diamonds. Now we are at 25%. It was odd. It was not a face card. It had a sharp edge, and there was a letter. If we take all of these into account, in an entire deck of cards, in the entire population, if you will, we were able to narrow it down to 1 card just by a simple blood sample. And that 1 card is the Ace of Diamonds. Now I hope you can see how useful and how brilliant, honestly, forensic biology is when it comes to blood samples and genetic profiles, because it can make the most impossible crime, let's say a few decades ago, and it makes it so possible nowadays to narrow out, yeah, they weren't blonde, or yeah, they didn't have blue eyes, but nowadays we don't even look at those characteristics. We put the 2-7, the 8-4, the whatever your genetic profile is into CODIS, and it sits there in CODIS until you are arrested or something along those lines, and you have to give a known standard. When you give a known standard, your genetic profile is put into CODIS, and if you previously committed a crime, CODIS just says, hey, that genetic profile you just put in matches the one from 10 years ago for this burglary. It makes it so much easier nowadays. In ISM, we have to create a product. That is something that helps our mentor, that aids them in some way, shape, or form. Originally, what my product was going to be was a crash course video, a sit-down kind of talk to the camera, and be a Khan Academy for forensic biology, because nowadays forensic biology is misconstrued due to the media, due to all these NCIS shows and Criminal Minds. It's nothing like how the media is presenting it, and that's the one thing my mentor had trouble with. He told me that, hey, it's going to be expensive and it's going to take time to actually do like an experiment or to try something in the sciencey field. There's an actual problem with the media saying there's like forensic biologists in cars with guns and hunting down the bad guy, when actually that's not what happens. Usually we're in the lab and we're only called into court to testify for the actual genetic profile and to state the facts. We're not allowed to give opinions or anything like that. And the media has really misconstrued that. Do you think you can make something to inform the public about what real forensic biology is? I said, yeah. And this was the original idea. And through many, many revisions, we came across an idea of, rather than teaching, we made it more interactive. So we thought, why not make a first-person sort of, I guess, simulation, if you will. If you've ever played a video game, you've played a first-person shooter. This would be a first-person forensic biology simulation. And what it would be was that we would get actors and have sets and put a helmet on one of the actors, put a camera on it so their hands were free, and they would go throughout the scene and you would be seeing through their point of view. And it was a very brilliant idea, and we were so close to doing it. We were actually 1 week away from shooting. I was finding actors, and then, as we all know, quarantine started. So I had to cancel everything and rethink some stuff, because the only alternative would be I filmed everything by myself and I would play every single character, and that was just not something that was viable. So we had to rethink again, and the issue was we had the script ready, 33 pages of science and jokes and a ton of inside Easter eggs that you need to find. We had all of that written, so we didn't want to throw it away. So we came up with my product that I finally was able to do, which was a choose your own adventure. And you see this graph on screen and you're probably thinking, I don't know what that means. Well, a choose your own adventure basically means you are given choices. When you choose something, you go down a different path. You know, I came across two roads that diverged in a yellow wood. I took the one less traveled, that whole thing. It's that principle of you have a choice. Do you want to go left or go right? The way you choose determines the outcome. And so you can think about it as going through different timelines. Let's say left, you face a bear, and right, you find a pond. Those are two different experiences. So that's what we were going to do with the first person video. Was at the end of the video there would be two options. You know, do you want to go analyze the broken glass or do you want to go talk to the victim? And by choosing, you would go down a different timeline, and sometimes the simulation would break due to your choices because you went against the rules. So we changed it so it was actually just in text format, where now people just click on the selection they want to do, and the text will show up and they have to read it and understand it. Hey, that's the lawyer talking, or hey, that's the victim, or stuff like that. And so it is upsetting that I was not able to do what I originally intended, but I do think, given the circumstances, that the product I did create was well worth it. And if you want to check it out and go down these different timelines for yourself, there is a link in the description to the actual choose your own adventure. Fair warning, there are many options where you can just end the game and you have to restart. That's just how life works. You go down one path, you can't choose the other. So, fair warning. But we were very happy that it worked out in the end, and I think it helps show people what true forensic biology is, because you go through different perspectives. First, you're someone in a waiting room trying out a simulation. Then you're in the simulation as the criminal, and then you jump to being the CSI. Then you jump to being the forensic biologist, and then you jump to being a jury member. You jump through perspectives, and whereas most people in crime scene analysis and the forensic fields don't see every portion of the actual process, like the CSIs, they only collect the evidence and send it off. They don't see what happens with the verdict or anything like that. With this, you're able to see the entire process from start to finish. I want to thank you all for taking, 30 some odd minutes out of your day just to watch my video. I hope you all stay safe. I hope that some of you find some enjoyment in my product. I worked many nights writing that script, and I tried to make it so that people who went to Clark or people who knew me would see some inside jokes. There were going to be many more in the video format. There were going to be Easter eggs hidden around, but again, times change, and so must I. Thank you so much, and good day.